Hi, my name is Charlize Norvell, and my research is called Is Brightening of Brain Spatial Detection of Microplastics. I was able to do my research at the University of California, Davis, within Dr. Newman's lab, and my mentor is Karina Burgos. Introduction. Plastics can be found everywhere from water bottles, hygiene products, utensils, tires, and cleaning products that we often use on a daily basis. And over time, these plastics degrade into microplastics. Microplastics are, to the naked eye, they're not visible, but they're go into a formation of micro and nanoscale, which can enter an organism's body through absorbization and helialization. Polystyrene, which is known as PS, and so this is a plastic that is single usable due to it not being able to fully degrade. And MALDI MSI helps detect the identification of polystyrene. Two key lipids that were analyzed in this study are phosphatidic acid, PA34-1. 34 represents the number of carbons, while the 1 represents the double bond, which is illustrated in the figure below on the far left. And so the main structure of phosphatidic acid is that it regulates synaptic vesicle fusion and membrane mobility, while the other lipid is singular myelin, which is SN34102. 34 represents the number of carbons, the 1 represents the singular double bond, and the O2 represents the oxygen containing two different modifications, which is illustrated in the figure below on the far right. And so the main functions of singular myelin is that it helps with myelin sheath formation and axon maturation. My hypothesis for the study was that polystyrene was gonna deregulate the PA341 and SN34102. And so I said that due to this deregulation, this would affect changes in synaptic and myelin sheath function. And I also expect that due to the polystyrene exposure, that it would mainly affect the hippocampus region of the rat brains that were used in the study. The materials and methods. So the materials used in the study were 6 Rister rats, which 3 were female and the other 3 were males. And so all 6 were about a year old and they were all lab grown. So they're lab grown to prevent any plastic exposure. So the way they ate and consumed things through metal containers and utensils. And so after the rat brains were killed, they were then their brains were then exposed to the microplastics and the brains were removed. And then afterwards, the brains were flash frozen. And then from there, the brain tissues were cryo-sectioned on average 10 to 20 microns. MALDI MSI, this is a very important tool that helped identify that polystyrene was a microplastic detected within these Ristar rats that were used in the study. And so after the rat brains that were dosed with the polystyrene were cryosection, they were then sprayed with the MP3 sprayer in order to preserve the tissue where it didn't allow too many ions nor information to be lost. And so after the tissues were sprayed, which is the matrix, they were then ready to be inserted into MALDI for ionization and cytotomics analyzation. And so with this, the instrument that was used was a UV laser. And so as the laser scanned across the tissue, analyzing the molecules, it then allows the molecules to go to a gaseous state. And so then this creates an ion plumb at each pixel, which can be, as a result, able to create an image, which is shown in the figure on the far right in the black and white for A and B. And so that shows 2 different ions images. And then after the ions go through MALDI, it then goes through timsTOF. And so this pretty much is another further identification that polystyrene is the microplastic within these rats. And so this creates a 1 over K0, which provides information about the ion structure. So as ions goes through the tubes, they fly through the tube flight, which heads towards the detector. And so as you can see in the figure on the below, the ions kind of are trapped in the tube, and as the gas flow goes against the electrical field. And so this gives us information to find regarding the final determination on the ions based off the speed in which it takes from the detector from TIMS using time of flight, which is tau. And so it's important to note that during this time period, that depending on the ion size and shape, so the larger they are, the more time it takes for them to go through the detector in the tube. And overall, TIMS TOF is a two-step verification that ensures the correct ID is given to each ion. And after the ions flow out of the TIMS TOF, they are then undergo the software called SKILLS, which is used by MALDI in order to map out the concentration of various ions throughout the experimental control groups in the brain. The last step within this study is known as H&E staining. And so H&E staining pretty much it pretty much helps get a better visual identification of cell morphology and the anatomical variation of the rat brains with the dose polystyrene standard. And after H&E staining, the slides were supposed to undergo through the microscope, but however, during my time in the lab, the microscope was not working. Therefore, no microscopic images were able to be processed. The results regarding the polystyrene section on the figure on the far left, this shows that as a result of using TIMS and TOF, it further identified the success of detecting polystyrene. And so you could see the highest peak, which is in black, shows that a fan-measured M/Z value of C11, indicating that a measurable polystyrene concentrated within the tissue from the just polystyrene rat brains. While the figure on the right, figure 2, this reveals ion mobility for ions composing polystyrene with different 1 over K0 values corresponding to mass to charge ratios. And so both findings on the slide further validate that polystyrene particles are trapped in ion lines and identified within the rat dose brains. Well, this slide shows the results of the lipid changes. So for lipid PA34-1, you could see there's a higher abundance within the controlled deionized water rat brains compared to the dose polystyrene rat brains. And this is the same case on the right in the figure 4 of lipid SN34102. And so as a result from the last slide regarding the lipids, you can see in this slide that dose 1.1 shows lipid PA34-1, while B dose 1.2 shows the polystyrene dose rat brains for lipid SN34102. And so with reduction of the PA lipid, this causes impaired neurotransmitter release, while for lipid SN, this may affect myelinization. And so both can resemble earlier neurodegenerative changes within the rats. This slide shows the neuromechanistic model. And so overall, this slide is like explaining visualization of lipid dysregulation, which alters brain chemistry. And so from the lipid, if you recall back to the lipid results, you saw how lipid PA had a lower abundance. And so with this is important because lipid PA regulates motor control and autonomic processing and sensory integration. And so disruptions in lipid PA association pathways could mirror earlier mechanisms observed in Parkinson's disease, where dopamine signaling is reduced due to loss of dopaminergic neurons and impaired receptor binding. And so this suggests that regarding polystyrene accumulation may indirectly contribute to neurochemical imbalances relevant to Parkinson-like dysfunctions. Similarly, for SN34102, downregulation as shown in the lipid results slide. And so this interferes from the myelin sheath integrity and signal transduction along the axon membranes. And so SM is a pretty important role for myelin due to insulating neurons and enhancing nerve conduction. So decreasing these levels could lead to slower and complete neural communication, which is often seen in multiple sclerosis, which is a big issue within the African American community. So both data from the results suggest that even low level polystyrene exposure can interfere with lipid metabolic necessary for maintaining a normal mineralization and neural protection. The strengths within my experiment was that Maldi was able to further detect that polystyrene was microplastics detected in the rat brains, and along with Tim's TOF, which is pretty good. While the main limitation of the study was that due to the microscope not working from after conducting H&E staining, no visualization of the cell anatomy of the rat brain was able to be shown, which is an issue. And then another limitation was that the only distributed statistical variation was the Boston research plots, which I include for the results. And so this limits the ability to determine statistical significance of the observed differences. And so as a result, if I were able to continue this research, future research, I would want to incorporate a t-test or a no-vote test in histology to further validate and confirm the findings that were concluded in this research. Further implication. So although this study was conducted on rat brains, further findings could be important because you can link it to the human brain, although further research will be needed conducted to further validate this. But rat brains are often used in research because their brains kind of resemble a human brain, which is pretty important. And so this can even provide more information regarding neuro environmental toxins regarding neurodegenerative processes and diseases. So in conclusion, this research provided evidence that polystyrene microplastic exposure leads to lipid deregulation in the rat brain, characterized by the low abundance in PA341 and SN34102 intensities, along with the high exposure within the polystyrene in the midbrain of those rat brains. And so these molecular alterations may disrupt neural signaling, myelization, which can potentially lead to environmental microplastic exposure to neurodegenerative mechanisms. In the future, I would like to see further research regarding combining molecular imaging, biochemical assays, and histology methods to be crucial to further understand the neurological impact and also to see the impact of microplastic exposure in the human body. Here is my slides of references, and thank you so much for listening to my oral presentation.